Population genetics studies distribution of genes in populations and how the frequencies of genes and genotypes are maintained or changed. Population genetics is important for genetic counseling, forensic medicine, and genetic screening. Today, we will cover 3 topics. First, we will look at gene and genotype frequencies. For a given locus, the gene frequency, also called allele frequency, measures the proportion of chromosomes that contain a specific allele. The genotype frequency measures the proportion of each genotype in a population. For example, a population of 100 individuals has been tested for a specific gene. The gene has 2 alleles, allele 1 and allele 2. Therefore, there are 3 possible genotypes, 11, 12, and 22. And here are the numbers. What are the gene frequencies? And what are the genotype frequencies? The genotype frequency is obtained by dividing the count for each genotype by the total number of individuals. The frequency of genotype 11, denoted F11, equals 49 divided by 100. That's 0.49. Using the same method, we can obtain F12 and F22. To calculate gene frequency of allele 1, denoted P, we first count the total number of allele 1 in the population. Please note, each individual with the 11 genotype has 2 copies of allele 1, and each heterozygote has 1 copy of allele 1. Because each diploid somatic cell contains 2 copies of each autosome, the total number of alleles is 200. Therefore, the gene frequency of allele 1, P, equals 2 times 49 plus 42 divided by 200. That's 0.7. Using the same method, we can obtain the gene frequency of allele 2, denoted Q. Q equals 0.3. As you may find, sum of genotype frequency F11 plus F12 plus F22 equals 1. And P plus Q also equals 1. Now, let's move to part 2. We will look at Hardy-Weinberg's equilibrium. If a population is large and individuals mate at random with respect to their genotypes at a locus, the population is said to be in Hardy-Weinberg equilibrium. It means that there's a constant and predictable relationship between genotype frequencies and gene frequencies. Hardy-Weinberg equilibrium requires the following conditions. First, mating in a given population is random. Second, the breeding population is large. Third, there's equal fertility and no selection. Fourth, no new mutation. Finally, there's no migration of alleles into and out of the population. If a locus has 2 alleles represented by capital A and a small a, the frequency of these 2 alleles are P and Q respectively. Then the Hardy-Weinberg equilibrium states the frequency of capital AA genotype would be P squared. The frequency of small AA genotype would be Q squared. The frequency of heterozygote AA would be 2pq. Application of the Hardy-Weinberg law is a high-yield subject for USMLE. First, let's look at application of Hardy-Weinberg equilibrium in autosomal recessive diseases. Suppose there's autosomal recessive disease where the frequency of the affected in the population is 1 over 10,000. If the population is in Hardy-Weinberg equilibrium, the genotype frequency of the affected Q squared would equal 1 over 10,000. The gene frequency of the recessive allele Q would then be the square root of Q squared, or square root of 1 over 10,000. That's 1 over 100. The carrier or heterozygote frequency, 2pq, is usually approximated as 2q because p is so close to 1. Therefore, carrier frequency is then 1 over 50. To sum it up, for autosomal recessive disease with population frequency of 1 over 10,000, the carrier frequency is about 1 over 50. A typical question is given in this way. They will give you a family history of a man or woman along with data from a general population and ask you the probability of their child being affected. For example, a student has a sibling with an autosomal recessive disease that is present at birth. If that student were to have a child with an unrelated partner selected at random from the general population, and the disease frequency in the general population is 1 over 10,000, how much is the probability of their child being affected? This type of question can be solved in 3 steps. The first step is to determine the probability of the student being a carrier. Based on the vignette, the student is not affected. Therefore, the student has 2/3 chance of being a carrier. Detailed calculation has been covered in the Patterns of Inheritance 1 lecture. Second step is to determine the chance of the partner being a carrier. We have already done that in the previous slide. Step 3 is to determine the probability of the child being affected. That equals probability of student being a carrier times probability of random individual being a carrier times probability of 2 carriers producing an affected child. The result is 1 over 300. Compare that to the probability that 2 unrelated individuals with no history of disease in their family have an affected child. That's 1 over 10,000. So the probability in this case is much higher. Second, let's look at gene frequencies related to sex chromosomes. Here is a typical question related to X-linked trait. If 1 in 10,000 males have hemophilia A, how much is the chance of a woman in this population being a carrier? We know hemophilia A is an X-linked recessive disease. Males have only 1 X chromosome. If the male's X chromosome has the factor A mutation, he will have hemophilia A. If his X chromosome doesn't have a mutation, he will not develop disease. Therefore, the gene frequency for the disease is obtained simply by counting the proportion of affected males in the population. Since 1 in 10,000 males have hemophilia A, the gene frequency for the disease Q is 1 over 10,000. Females have two X chromosomes, so they must inherit two copies of mutated chromosomes to develop the disease. Gene frequencies are similar in males and females, so we can use the gene frequency estimated in the males to predict the genotype frequencies in females. If the population is in HardyWeinberg equilibrium, the frequency of the affected females is given by Q squared. The frequency of female heterozygote is given by 2pq, which is 1 over 5,000. Now, let's move to part 3. We will look at factors causing genetic variations in population. Although human populations are typically in Hardy-Weinberg equilibrium for most loci, deviation from equilibrium do occur. Factors responsible for genetic variations in population include mutation, natural selection, genetic drift, and gene flow. Here, mutation refers to new mutation. We will look further at natural selection, genetic drift, and gene flow. Natural selection acts on genetic variation, increasing the frequencies of genes that promote survival or fertility, and reduce the frequencies of genes that reduce fitness. For example, sickle cell disease, thalassemia, G6PD deficiency provide resistance to malaria. This mutation increases reproductive fitness of carriers. This is an example of results from negative selection. The first generation begins with two alleles with equal allele frequencies. If natural selection against AA genotype, people with AA genotype will not reproduce. As a result, allele frequency Q will be reduced to 1/3 in the second generation. If selection continues on the third generation, the Q will be further reduced to 1/4. If selection goes on, AA genotype will eventually disappear. On the other hand, if natural selection favors one particular genotype, that population will increase. Sickle cell disease is an example of deviation from Hardy-Weinberg equilibrium because of heterozygote advantage. Here, A represents normal hemoglobin allele, S represents sickle cell allele. These are actual data. Based on this data, we can easily obtain P and Q. Based on Hardy-Weinberg law, we can obtain frequencies for 3 genotypes and the number of people with each genotype. As shown in this table, as you can see, actual number for heterozygotes is much higher than predicted. Homozygous wild-type individuals are subject to selection by malaria. Homozygous mutant individuals are subject to selection by anemia. Heterozygotes have the greatest reproductive fitness and serve as a reservoir for the mutant allele. Genetic drift refers to gene frequency change caused by finite population size. This change can be quite rapid in very small populations. Genetic drift helps to explain why some genetic diseases are unusually common in small isolated populations. For example, more cases of the rare recessive Ellis-Van-Creveld syndrome are found in the old order Amish than in the rest of the world. People with this condition have short rib, heart malformation, and polydactyly, as shown here. Here are some rare recessive disorders common in some populations. For example, congenital nephrotic syndrome is commonly seen in Finns. Ellis-Van-Creveld syndrome is commonly seen in Amish, as we have discussed earlier. Tay-Sachs disease and Gaucher's disease are commonly seen in Ashkenazi Jews. As shown here, in a large population with random mating, large fluctuations of gene frequency are unlikely. In a small population, gene frequency can change rapidly from one generation to the next. For example, if only AA individuals participate in mating by chance in a small village, children from the village are all AA. The frequency of the small a allele is low in the initial population, but a small subset in which one individual is heterozygote is removed from the large population and founds a new population. The frequency of the small a is markedly higher in the new population due to its relatively high frequency in the founders. This is called founder effect. Gene flow refers to exchange of genes among populations. Because of gene flow, populations located close to one another often tend to have similar gene frequencies. For example, frequency of sickle cell disease is lower in African Americans, in part because of gene flow from neighboring populations that do not carry disease-causing mutation. In addition, the heterozygote advantage for sickle cell mutation has disappeared because malaria has been rare in North America.